Hi, everyone. It's my pleasure today to be here to introduce Dr. Edward Fox from Stanford. Edward's going to be talking about genetic variants in brain health and neurodegeneration. Edward, great to have you here today.
Thank you for the invitation to present our work to you. As Nikki mentioned, I was going to, can you hear me, Nikki, by the way, just to double check?
Yes, we can.
So as Nikki mentioned, I was going to talk briefly about some of our work on genetic variation and how we link that to neurodegenerative diseases. So as all of you are aware, there's an increasing number of GWAS studies on neurodegenerative diseases. And one of the outputs of these, or the main output, is we have an ever-increasing list of candidates of risk loci. And for us, we've had a long-term interest in trying to link those loci to pathological markers or disease progression, and basically understanding the disease. Now, there have been a number of challenges in this, and just very briefly to set up the problem, I'm going to show how we've addressed. So are really two big problems. One is most GWAS SNPs tend to be non-coding, so it's not that easy to necessarily interpret what their function might be in the cell and how they might be playing a role. And then secondly, there's a larger problem is we're not always sure when from GWAS studies what the candidate actually is. Usually what is actually inherited are blocks of SNPs rather than individual SNPs. So even though we'll have in a GWAS study a leading candidate, we often have a large number of co-inherited SNPs in a linked region. And it can be often difficult to understand which one of these you should be focusing on for functional validation. So what I'm going to show you here is how we've adapted our experimental and computational pipeline to try to address these issues, to focus on the non-coding SNPs, which are the vast majority of hits in these studies, and then how we whittle down to which we think are the most likely to be the functionally important ones driving the disease progression or the phenotype that it's correlated with. What I said, what I'm going to show is the experimental and computational framework and how we've dovetailed those to try to narrow down the candidates. And then our underlying assumption of all of this work, just to be clear from upfront, is that we expect that non-coding SNPs exert their function through a regulatory effect. So while they're not directly changing protein coding, we presume that they're affecting the expression of certain genes through their action. And then our approach has been to, rather than focus on disease phenotype specifically, we wanted to build a map of how we wanted to layer on epigenetic annotations on top of the GWAS studies to try refine our selection criteria. And really the first level of that was to build a map, a kind of a single cell map of certain regions of interest of human brain in a normal human brain. And the reason we focused on normal brain was that we want to see what the baseline for how the genome and the epigenome was behaving in these samples. And we did this with, as I'll show you in a second, through a TAC-seq and then 3D interaction, chromatin interactions. And then on top of that, we then try to layer on a prediction of when we've narrowed down our candidates, how can we understand exactly what or can we make an exact experimental prediction of what the effect is at the genome and epigenome level? And again, our first step was to build essentially a single cell map of brain regions and their epigenetic landscape. So we selected a number of disease-relevant brain regions, and then we applied a single cell TAC-seq, which, just for anyone not familiar, what we're really looking at is what parts of the genome are active or open. So what parts are interacting and what parts are regulatory elements binding to. And I won't show too many of these, but just to give you an idea of the idea. So for example, if we're looking at a dopamine receptor, which we expect to be, relevant we expect to be active in certain regions, we can see we have more open chromatin in the regions where we expect it to be active. And then in regions where we wouldn't expect it to be active, it's more silent. You can see here clearly in the substantia nigra, we have a more open chromatin for this particular gene. And then similar as we go through this potassium channel, for example, in certain areas of brain, certain subtypes, you see it's more open than in others. And this allows us to assay which areas of the chromatin are of the genome are actually we believe in regulatory roles. To get into a lot, what we end up basically is a single cell map. And what I'm showing here is what you've probably seen from a lot of publications, is a UMAP. It's just basically all the cells from cognitively normal brains and mapped into their different genome types and their cell subtypes. And the advantage of this technique is that it lets us get individual resolution for particular subtypes. And we would expect that a particular risk loci will be exerting effect only in the subtypes of cells. So for example, a neurodegenerative risk loci may only be active or may only be exerting its effect or its risk effect in a subset of neuron types. So that's why this level of resolution, where we're actually down at the single cell level, allows us to tease out effects that are in specific vulnerable cell populations. I'll give you first, just to show you the pipeline, kind of a schematic version of how this works, and then I'll go through a specific example. So typically from a GWAS study, what you'll have is for a particular chromosomal location, you'll have a hit that looks particularly intriguing. It has a high linkage to the phenotype you're correlating with, in this case neurodegeneration, and then looks like a plausible candidate based on prior annotation from the literature. Now, in truth, as I said, when you look into the data, you'll actually have a large number of SNPs that are co-inherited frequently with this lead candidate. So really, it can be any one of these. You're basing your best assumption, this guy, based on prior literature. So what we do is then layer on some experimental evidence about the epigenome. So we look at what layers are open. So these are the chromatin peaks I was talking about before. And then we also look at what regions of the genome that are open are also behaving like regulatory regions. And the idea here is being that regulatory regions will interact with other regulatory regions. They come together and DNA loops, essentially. So by doing this approach, we can narrow down from our original cohort of candidates. We can essentially narrow down to the ones that are most likely, so the ones marked in red here. So they have the phenotype that they're in open in chromatin, so the chromatin is active, and then the chromatin is behaving in a way that we would expect it to have a regulatory effect. So to show you this for a specific example, so this is the BIN1 locus, which is a risk locus for Alzheimer's disease. It's well characterized, but again, we'll show here, even using our pipeline, we think we can actually add annotation, not just to novel loci, but even to loci that are believed to be quite well characterized. So what we have along here is where we've broken the data down into the individual cell types, the major cell types within certain brain regions. And what you can see is for BIN1, so this is our lead SNP from a GWAS study. We have 2 lead SNPs. But when we actually look in the data again, we see that there's a large number, there's over 100 SNPs that actually are plausibly linked as the 2 lead candidates. So using the chromatin profiles, we can see there's actually in microglia, which is the cell type we believe to be most vulnerable in this risk loci, one of the candidate SNPs maps to open chromatin, but so do others. So it kind of leads us, as we proceed down through this, by layering on the epigenome annotation, that we have multiple candidates actually satisfy our criteria. And if we go to the next level, if I can show you again, we have our starting candidates. We filter through. If we then add in our epigenetic interactions using what have active promoter and enhancer markers, and then which chromatin peaks are actually co-occurring in individual samples, we can see that our most likely candidate is actually this SNP here, rather than one of the original leads. So we can show that we can take this and make a prediction that we actually believe that this seems to be the most active of the linked SNPs. This is the one that is most active. And what we can do is, this is the machine learning aspect that I won't go into so in depth into, is we can then genome-wide predict what the effect of this alteration is likely to be. And for this particular locus, what we see is what's actually happening is we're in individuals who are contained or carry this risk loci, they're actually losing the function of a key transcription factor binding site regulating BIN1. If I go back, sorry, briefly, what you can see is that this site, when we lose this, when this SNP is the risk allele, we lose all these regulatory interactions, both from the open chromatin and then within the active transcription or enhancer and promoter regions. So basically, we've lost this level of this, what we see in normal brain as being the key and most active enhancer regulatory element, is actually its activity is almost completely ablated in the individuals who carry this risk loci. So basically what we end up with at the end, as I said, is we end up with a list of predictions of which are the most likely candidates that we should go on to validate. And this is where our current work is at the moment. So we're trying to essentially, we take GWAS studies, we build a map of normal brain, of how it interacts and behaves at an epigenetic level, layer that on top of the GWAS data. We've come up with a list of what we believe are the top candidates based on experimental annotation. And then we take that prediction and we feed it into, and this is our ongoing work, we do massively parallel reporter assays and scarlet genome editing using CRISPR-Cas9 to see if we can validate our predictions. That's our current ongoing work at the moment. And just finally, I'd like to thank Ryan Courses, who was the lead on this study and has now taken a faculty position at UCSF, and then our team lead, Tom Montine, who couldn't be here this morning. Yeah. Thank you. If you have any questions, I'd be happy to take them.
Edward, thank you so much. I'm going to just monitor the chat here and see if we have any questions from our audience.
Of course. Thank you, Nikki. And thank you for the opportunity again to present. This was I hope I didn't go too quickly in covering our work. And one last thing, while we're waiting for questions. If anyone does have candidates that are interested in looking through the annotation, we've published our pipeline here, and we can help if people have particular loci they're interested in looking at what we would predict the true risk loci is.
So Edward, this might be, sort of it dovetails into your work, the question that I had, and I think of, so our group is examining the role of various microbes in Alzheimer's disease, neurodegenerative processes. So what's fascinating about your work is the potential marriage, that can take place between identifying, certain epigenetic traits or characteristics that can be then potentially correlated or better understood in the context of a larger picture. One of the fascinating things is that it appears that the ApoE4 variant, or individuals with two copies, certain groups have examined that particular genetic trait in reference to vulnerability to infections. And it appears that there's a relationship there between the genetic trait and the immune system. Have you guys thought at all about the application of some of what you're doing from an immunological standpoint?
Not outside the central nervous system, but yeah, that would be very intriguing or interesting to look at. So essentially, any phenotype that can be correlated with a GWAS could be mined down. And I think kind of a more holistic approach to, we're very central nervous system centric, but we are looking at more peripheral biomarkers and peripheral phenotypes that are indicative of disease course and risk. Is definitely something that could be incorporated into this kind of pipeline to see what are the underlying genetic risk loci that add to or undermine those phenotypes.
Wonderful. And we do have a question. Are epigenetic characteristics protecting cognitive reserve and decrease on Alzheimer's disease similar?
I'm not sure they're similar, but there are definitely contributions in both directions. I think if that's get at the question, so I'm not sure. I would say they're similar, but they are both active. So there are resilience factors we're looking for and resistance factors that we believe would be essentially independent markers of cognitive reserve and cognitive resilience to disease.
Thank you so much. And Lavinia asked another question. Which classes of variants are mostly represented?
So for what we're looking at, it's mainly single nucleotide polymorphisms is major for our pipeline. Generally, the effect size of larger structural variants is somewhat easier to predict because they're more catastrophic in their impact and they're less subtle. So our pipeline is more skewed towards single nucleotide polymorphisms.
Edward, thank you so much. We're about to begin our next session, but we really appreciate your presentation. It was very interesting and informative.
Thank you. And thank you again for the opportunity to present.
Absolutely.